I'll try to get up to date this weekend. Yeah, my research student was going out of town for like 3 weeks, starting today. So she had all these experiments going on in the lab, and so I was assisting her. Hence, I got a little bit backtracked, but I am working through your experimental design sheets. I am noticing some like recurring mistakes that I'll discuss later today, but if you don't have a grade for it, you will have a grade for it by the end of day today, because I want you to know what you've done incorrectly before you take the quiz this week, since there'll be questions like that on the quiz. But yeah, most of you, the biggest issue that I'm seeing is with the hypotheses. You want to make sure that your hypothesis has your independent and dependent variable clearly identified in that statement, and also that it is a testable statement, right? So the hypothesis has to be something testable. It can't be just a statement. I've had people that have written statements like black mulberry and saffron will show anti-cancer activity at 100 microgram per microliter. You don't know that exact concentration. That's why you're going to do the experiment. It has to be something very specific, or not specific, but rather general, and it has to be a testable statement. Also, try to think about, since there are those two supplements, go through the module and try to think about which one do you think is going to work better, maybe, or which one may cause a growth arrest versus a death in the cell, and incorporate that into that hypothesis to make, to help us see what it is that you're going to be looking at. What is it that you're going to call your dependent variable? And making that hypothesis clear in that way will also help you figure out those variables too. Some of you are still writing both viability and death in the dependent variable, but the dependent variable should be one of the two. You decide which one it is that you're going to be focusing on for the case study, okay? Does that make sense? Yes, no?
Yes.
Okay. Also, today, obviously, you start working with your group. So one of the first things that you will do when you get into your groups is go over your hypotheses and develop a unified hypothesis that you will be working with together as the group. So you will have a chance to kind of, remake that hypothesis today, and that will be part of your assignment today as well. Okay, so I'm going to go ahead and share my screen so we can start talking about our module this week and what we are going to be focusing on. So this week we are doing cell viability and let me close this out. Cell viability and morphology analysis. So we talked a little bit about the morphology stuff last week, and that's why I had you guys take that quiz last week, because I wanted you to be a little bit familiar with the different parts of the cell and the way the cells may show when they're going through different stages of maybe cell cycle or different types of health conditions, and how that would show. So today we are going to actually be visualizing those things inside an actual image from one of those treatments that are part of the case study. So today, looking at that first sheet. So if you look at the module today, it's going to give you your objectives, just like always. Our main goal today is to understand different ways that we can look at the health of the cell and the viability of the cell in a molecular biology lab. We are going to do that by looking at several different kind of parameters of cell viability and cell health. One of them is simply looking at whether they're alive or dead, right? So looking at markers for different types of cell death. We again talked about some of this last week, and we'll review them today as well. So cell death can occur through one of three different ways, usually when we are treating them with some kind of a drug or a compound. And those are apoptosis, necrosis, and autophagy. So today we're going to try to, as one of our observations, you can try to observe the cells and see how the cells are dying. The ones that are dying, how are they dying? Are they dying through programmed cell death? Are they dying through this explosive necrosis? Or are they dying because of a starvation response, whether it is a perceived or a real starvation response of the cell? The second thing that we're going to be looking at is for the cells that are there and that are alive, how healthy are they? Now, there are different ways that we can look at that. Some are more qualitative and others are more quantitative. So we already did a little bit of observations in class last week with the trypan blue exclusion, right? Where you trypsinize the cells in your culture, you add trypan blue exclusion reagent to it, trypan blue reagent to it, and then you count the number of clear versus blue cells. When you do that, which ones are going to be your living cells? Are they your clear or the blue?
Clear.
Clear. Why not the blue ones?
The blue ones didn't have the ability to not let the dye in the cell.
So the blue ones are the ones that are letting the dye in, which indicates what?
Their cell membrane is permeable.
Exactly, that they have lost, the integrity of the membrane. It's become more permeable, so more things can get in. So that is why you know that those cells are dying or are really stressed and are on their way out. The second thing that we can look at is a crystal violet assay. Now, a crystal violet growth assay, relative growth assay, or viability assay simply uses the dye as a way to look at how many cells are on your plate or cover slip or dish. So this is, different than trypan blue. Here, the blue dye indicates actual cells. So in this case, we usually stain the cell with the crystal violet dye at different time points, starting at the 0 hour, usually going all the way up to 96 hours sometime. And then we can observe them under the microscope, but more importantly, we can resolubilize this dye back and we can look at the response of the cell culture over time. If you had the cells grow over time, you would expect to see more and more blue in that resolubilized reagent. If your cells died over time, you would see less and less blue in that case. So this is going to show, the more blue is there, the more cells are there. However, the backdrop of this one is that it's not really looking at the health of the cell. It's just kind of looking at, are the cells there or not? So just kind of mostly living, and maybe some that are on their way out, but they're still attached cells. The last thing that we can look at, and this is the most quantitative and accurate depiction of the cell health, is some kind of a metabolic assay like MTT or XTT assays. These assays specifically look at either mitochondrial enzymes or other metabolic pathways to look at, maybe ATP production or ATP content inside the cell, or just simply activity of the cell. So in general, the more active a cell is, the more healthy it is. So in this way, you can actually not only look at how many cells are there, but how metabolically active they are. Are they stressed? Are they, healthy? And you can kind of monitor their overall health as well, in addition to just looking at their relative number or relative growth. So today in class, however, majority of the data that you will collect is going to be from trypan blue images. We will talk about how to interpret the crystal violet or MTT analyses, but you won't actually get any data from them yet. I will give you data from there at another time when we are kind of combining all our data together. So today, like I said, you're going to be determining cell viability using trypan blue images. You are also going to be looking at how you would then interpret those images with appropriate statistical analyses, and how would you present your findings in the form of a scientific figure. So for that, I will have like a mini Excel figure making, PowerPoint figure making workshop towards the end of the class, after you've had some chance to collect the data. Okay. So this particular page kind of talks about different ways that we can look at cell viability, that we can look at cell health, kind of overall information about that. Some of these things we are going to be looking at today, like we are going to be looking at cell numbers. We are going to be looking at cell viability analyses, data like the metabolic activity with MTT and other things. Other stuff we are not going to be looking at today, but we will be working on them over the next couple of weeks as we talk about DNA analysis with DNA content and, fluorescent assisted cell sorting later on in the semester. There is this PowerPoint down here that goes through all these different kind of techniques that are used to assess cell viability, cell health, and cell cycle. And there is a little kind of presentation by me, a lecture of that by me underneath it that you can watch to learn more about it. I'm going to go about over some of it in a second as well. I also, even though we weren't doing the lab face-to-face, I still kept your protocols and the flowchart there for you to see, because I wanted you to be familiar with the process with which we would have collected these cells and we would have observed them in the lab as well. So you can kind of have a little look at that and see how you would have done that. So for example, the first one is with the trypan blue exclusion assay. It kind of walks you through how we would have trypsinized the cells, just like we have done a couple of weeks ago when we did our aseptic technique. And then at the end of the day, instead of plating those cells, we are taking our suspension and we are adding the trypan blue stain to it in order to observe them with a hemocytometer and look at the number of clear versus number of blue cells. Remember, when you're looking at these images, they may not be, they're not going to be necessarily this bright blue. Many times they show up as these very faint blue cells, and those are dying cells. Also with the clear cells, you will see when you get actual images from the microscope, sometimes they have this kind of a glassy look. They look almost like a gray cell. They don't look like a clear cell. But those cells are clear when they have that gray tone to them and not the blue tone to them. And then for the morphology analysis, obviously we are going to be looking at stain slide images. This kind of walks you through the way you would have stained the cells, just like we did last week in class. And then this time you will be provided with an image to make your observations with. And then it talks about what are some of the ways that we can make observations. We're actually going to make different observations than the ones that are listed here, but death is one of the criteria that we'll be looking at, and you can look at that through this as well. Okay. The last thing that I have in here is calculating IC50 of a given drug. Does anyone know what an IC50 is? Especially those who've taken chemistry, I think. I can't see the chat, so I don't know if somebody is writing in the chat. So IC50 of a drug is essentially the concentration at which 50% of the cells are dying or are affected by that drug. So when we are doing IC50, calculating the IC50 of a drug, many times what we are running is a crystal violet or an MTT assay with a range of concentrations, maybe a serial dilution of the concentration of the drugs from really low to super high. And then we observe them over time, and we calculate at what concentration 50% of the cells die in a given time point, and that's considered the IC50. So this particular video is going to walk you through how we would set up that experiment, how we would make those observations, and how we would interpret the actual graph at the end of the day. So if I switch off this, I'm just going to show you one little thing from there. So it's going to kind of walk you through the analysis, how you would do it, everything. And then it's going to show you, look at the graph there. So it's going to show you, oh no, what did I do? I want to pause it, man. Guess I don't want to listen to me. That's so the actual graph part, he goes over really quickly, but basically it talks about how you would take your concentration on the x-axis, the log of the concentration, and the cell viability on the y-axis, and you would take a look at the 50% when 50% of the cells are dead. And that's going to be equivalent to the concentration of the drug that is needed to kill 50% of the cells. So that is something that I would like you to be able to do, looking at a graph of something like this and deciding where the graph can be presented different ways. It doesn't have to show it in just that format with the log of the concentration. It could be simply with the concentration on the x-axis and the percent viability on the y. You should be able to tell the 50% point and the concentration of the drug, determine the concentration of the drug. Lastly, it has your assignments for this week. You have your cell viability analysis assignment, which you will do as a group. And then you have the experimental design and the viability post-lab quiz that you will do individually. And we'll go over those in just a minute. Any questions up till now?
I just clicked on the cell viability analysis assignment and it said it had been deleted.
It says the same for me. Are you kidding me? What the heck? That's strange. It's not supposed to be deleted. It says Zoom session 3 cell viability. You see that? Are you kidding me? Okay, let's fix that. Let us fix that. It was under modules, right? So what happens when I click on this? Did you click on this one?
I clicked the one within the...
Within the, so I think within that, it must have gotten a broken link or something. Let's fix it before I go forward. I want students to be like completely lost. Okay, mister. Don't like us? Fine. Sometimes the links get broken. If I edit the assignments too much, and I've been kind of editing that one a little bit. So I already had the assignment in place, but then I edited it a few times to update it for summer. Try it now. Oh, so now it's working. Cool.
Yeah, it's
Is
working.
it working for you as well?
Yes.
Perfect. Okay, any questions before we go forward? Okay. Okay, so let's go up here for a second and kind of go over a little bit of this. I'm going to actually mute this one. You can listen to it later in more detail. I'm just going to point out a few things in here. So this is going to kind of go over the cell cycle stuff that we did a little bit last week and talk about how the health is going to be changing with different drugs and what happens when the cells are in growth arrest or senescent, or they're actively dividing, depending on what it is. And it's gonna then go over some assays. I'm trying to get to it. So one of the assays that it's going to talk about is crystal violet proliferation assay. Now, with the crystal violet stain, you can look at many different things with that stain because it kind of stains those cells very nice and clean. It's quick, it's very low cost. So it's an easy way to kind of look at overall how many cells are there, how many colonies are there, things like that. Some of the ways that you will see the data would simply be some relative growth graphs or line graphs where it shows growth of cells over time. Otherwise, other times you may see the data in one of these formats. So I thought I would show you some examples. So one thing you may see is images of cells that are grown in culture, either individual cells or colonies of cells. So especially cancer cells, we can do this assay, which is called a tumorogenicity assay, where we embed the cell in kind of a little agar medium. So it's a jelly-like medium on the bottom of the plates. So they don't really have a place to attach to. In that case, they kind of grow in little balls instead of growing in a flat sheet as you typically see. Each ball is considered a colony. Now, normal cells are not going to be able to make those colonies. They will just die out in that agar. But cancer cells will make colonies. So especially for looking at how aggressive a cancer is, how tumorigenic a cell line is, we will run this experiment called colony formation assay or tumorogenicity assay. In that case, crystal violet staining is a very easy way to stain those colonies of cells, and then to observe them under the microscope and count how many are there, how big they are, all that good stuff. And so here is an example of that type of data where they've done a tumorogenicity assay with the increasing concentration of the drug, and they've counted the colonies, and they are showing that representation, how over, with increasing concentration of the drug, they found less and less colonies. So here you see, in the untreated and with the zero concentration of drug, you have a lot of colonies all throughout. In the low concentration, you still have some colonies, and some are really large as well, but they are significantly reduced. They're beginning to show significance. And then with each additional concentration, with increasing concentration, you see less and less colonies. So this particular drug, which we're showing the effectiveness in this way, showing that you can reduce its tumorogenicity of a tumor or a cell line, of the cells by adding this drug to them, giving them this drug. This particular image, if you look right here with the ABCD, is what we call a multi-panel figure, a kind of like the figure that you will be making today as well, or this week as well. A multi-panel figure is where you have multiple panels of data, multiple kinds of data combined together in a single figure, all leading to the same final conclusion. So here, this panel C is showing that tumorogenicity is reduced, less tumors will be formed in the presence of this drug, and they'll be smaller in size. If you look at this first panel, it shows that if you just have cells in culture, as you provide this drug, it does show decrease in cell viability. So you have less and less cell survival over the course of treatment with increasing concentration of the drug. So, it has different ways to show the same conclusion. right here, this is just showing you how crystal violet itself shows increasing color with increasing concentration of things to bind to. So this doesn't have any cells if you look at that. It's just showing the optical density of the dye, optical density due to color. And it's showing that as you increase that blue dye concentration, you have higher and higher absorbance. And that's all that that particular one is showing. And then in panel D, it has an absolute cell count where it shows the more cells you have, the more blue you'll have. And you can look at that as well right there, right? So, and you can even get absolute cell count or estimate absolute cell number in a dish by looking at how blue it is in crystal violet. However, to do that, what you need to do is first make a standard curve where you take different known numbers of cells in a plate, stain them with crystal violet, and create a crystal violet, absorbance where on the y-axis and the cell number on the x-axis curve, which would be a standard curve to compare against.
I have a question about the
Yes.
Optical density of the dye. What was the point of that one again? I didn't really understand that. What's the point of that graph?
The point of that graph was actually it's kind of proof of concept graph, where they're saying that if we attach this drug to the dye and we are using increasing concentration of the drug, hence increasing concentration of the dye, the optical density will increase. That's all that it's showing at that point. So this is just, there's no cells in this culture. You see that right there, just empty things. It's just showing that the more blue you have, the more absorbance you have.
Okay. Mm-hmm.
This is actually a figure from one of the first papers with crystal violet, where they were kind of showing the applicability of that in culture and what different things you can do with it. Okay, so then you have your trypan blue exclusion, which you all already know about with the clear and the blue. Here you can see those glassy cells that I was talking about, right? They have a little shape to them, but they're clear. Those are not blue cells. And you will have some cultures today, some images today that show cells like that. And that was a little bit confusing yesterday for some students as well. But these glassy looking cells are clear cells. And these with either a light shade of blue or dark blue are the dead cells. So it's not like completely colorless. Now, both with the crystal violet and with any of the metabolic assays like the XTT and MTT, one way that we can look at them is by looking at the cells over time and looking at either metabolic activity or just simple changes in cell number. So for a metabolic assay, usually those are short term. You look at them sometime usually between 4 to 12 hours. For cell viability, you look at cells long term. So you look at usually 24, 48, 72, up to 96 hours, and you watch over time how the cells are growing. But if you're just looking at metabolic activity and the health of cells, you're only going to do short time frame. So here you're seeing you have plated, in this case, they have plated different number of cells at the beginning time point, and they're looking at their metabolic activity over time. And what you're seeing on the y-axis is the absorbance at 450, which is indicative of, that activity. The higher the absorbance, the more metabolically active cells are. Now, each cell line will have different level of basal metabolic activity that you need to know before you can compare it to any treatment. So we always do one experiment just like this. So we have the basal level of metabolic activity of the cells before we actually run our actual experiment where we have the different treatment groups with same number of starting cells to work with. the next thing that I will show you is another thing that we will work with later in the semester, but I wanted to show you this data now because many of your primary literature articles that you will see in between do have this data. So this technique is called fluorescence activated cell sorting or fluorescent assisted cell sorting or FACS analysis for short. FACS cell cycle analysis looks at not the health of the cell so much as the state of the cell in the cell cycle. So what it is saying is that these cells that are living, that are on your plate, what exactly is happening to them? Are they in quiescent or G1 state? Are they in synthesis phase? Are they in G2M phase? So where exactly do they stand in the cell cycle? To do this, we get our cells, trypsinize them at the end of our time points in the various treatments, and we fix them using ethanol. So similar to the fixatives we used last week, which were alcohol-based, in this case, we use alcohol as well. And so the alcohol in the suspension, what it does is that it fixes all the cells in the state that they are in so that they can be preserved to be examined later. They'll still have all their membranes intact. They'll have all the stuff inside intact in that state. We then go through a couple of steps of membrane permeabilization to kind of make the membranes a little bit more permeable. And we add a dye called propidium iodide, which is an intercalator. So it intercalates into the double-stranded DNA strands and stains the DNA specifically, obviously in the nucleus. That's where majority of that DNA is gonna be. We then make it go through the flow cytometer, which essentially takes a very narrow stream of the fluid so that it can look at every single cell as it passes through. It passes a laser through it to examine the fluorescence that's coming off that cell with the propidium iodide staining. And what you end up with is a scatter plot with individual dots for representing every single cell, and a plot that looks something like this that shows the number of cells at various level of fluorescence. So using that, we can estimate the number of cells that are in either 2N DNA content or G0, G1 phase or G2M phase. So the first peak doesn't have to be the higher one. That's a typical graph if the cells are in a normal, healthy state. Usually you'll see a lot of the cells in G0, G1 phase. You'll see some cells in S phase, and then you'll have a second peak at twice the fluorescence of the first, indicating the G2M phase cells. So that's a flow cytometer cell cycle analysis. If you have cells that are dying or that are fragmented, have fragmented DNA, they will fall below this, the 2N part. And many times you'll see them kind of accumulate in that really small, peak right here that you are seeing in here. These are your apoptotic cells. So they'll have fluorescence below your 2N content. Questions about this graph? Okay,
No.
there's a quiet. Yes,
No, I said no questions.
okay. Okay. So like I said, we will actually be looking at a lot of data in primary literature that has these as well, but we are also going to see some example data to work with in a couple of weeks when we do our DNA module. Okay, so let's look at our assignment a little bit before I divide you up into your groups so you can begin to take your initial observations. So you have an assignment file right up here. Oh my, it's right here. I think I may have it open already. No, I don't. I will open one. Okay, so this is your assignment file. If you open it up, you're going to write the aim of your experiment. Now, today, your aim is going to be different than last week, right? You have a particular goal today, that is to observe the viability of the cells in some way. So that's going to be what you will write in your aim. That's the focus, is the viability of your cells in the different conditions. For your hypothesis, you're gonna go through your hypothesis, individual hypothesis as a group, and generate a nice, clean, testable hypothesis, testable statement for your hypothesis, encompassing everybody's ideas into a single statement. And then you will have the data that you will be working with. So it tells you that you're going to be looking at primarily the trypan blue exclusion assay to determine the cell viability in response to the natural products that you're using. The first thing that you're going to be doing is what we call a relative growth analysis. So what I would like you to do, by the way, when you first get into your groups, is one of you can share their screen and open this up so you can kind of work on these things together. Also make, download this particular file and make it a shared file as a Google Drive file or whatever between the four of you. So every person can input their own data in the sheet. That makes it a lot easier for you to collaborate and work together. And having that screen open between you guys to look at the data together. There are some parts of data that you look at together and some parts individual. But for the relative growth especially, you're supposed to look at it together. So you can kind of work on it together with one person opening those slides on the screen and everyone then giving their ideas about what it is that you think it is. So the first one is what we call relative growth analysis. So in your assignment, you have this relative growth data file. So I'm going to show you that. You will open this up, and what you will do is you will look at the images that are provided to determine the confluency of your culture at the beginning and at the end. These are taken at really low magnification, so you see lots of cells, a bird's eye view of the culture, and what you can determine, and they're stained in blue, so that makes it even easier, right? In culture, normally we look at confluence when they're still unstained, so it makes it sometimes harder. This time, I've kept them stained, so it should be easy for you to determine what percentage of the image is covered in blue and what is clear. So the blue in this case is the cells. This is going to be used to determine the confluence of your culture. So you will look at this and you will say, okay, this is, you decide amongst yourself, everyone gives their answer, and then you write your answer, whether it's 50%, 60%, whatever. And then you look at the end and you look at that particular growth and you say, okay, there's only a little bit of white left. So this is 95% confluent or 100% confluent, whatever you want to call it. And those are what you will write in this first chart, right? All these charts are made so that you can simply click on this little square, copy it and paste it into Excel, and it will become an Excel table, no problem. There will be no issues in formatting, and you can continue working in there. So for the start and ending confluency, that's what you're writing in. And then for the relative growth, what you will do is you will divide the ending confluency with the start confluency. So you're not going to write this formula for all of yours. You're just writing your answer. This is just to say that in Excel, that's the formula you would write. You would write equal sign, ending confluency divided by starting confluency, and you would get a number. That number shows you how the cells grew over the course of the treatment. If the number is 1, that means the cells neither died nor grew. They remained overall stable in the cell number. If the number is above 1, that indicates the cells were dividing and now there are more cells, there's more coverage of the image than it was at the start of the treatment. If your number is below 1, that means the cells were dying and were not getting replaced. So there was some either growth arrest or cell death happening in that culture. Okay. So in this example, the number is 2. So what does that mean?
Growth.
Growth, exactly. There are twice as many cells now as when they started. So that's what you would do for this one. And then based on those results, you are going to write a couple of statements about whether the hypothesis was accepted or rejected and explain why your rationale behind your answer, basically. Okay. Then we get to the second part of your work, which is your trypan blue exclusion analysis. Again, you are provided with some raw images to analyze. Now, in this case, we are not doing a hemocytometer, but rather just a simple image, because it's basically a square worth of image that comes in your picture. So they are not exactly, you will notice they're not exactly the way you think they would be. They're just images. You're counting every single cell in here. So for example, in this particular image, you know, there's one, maybe two blue cells, everything else is clear, and then you'll count all the clear cells. There are 3 slides for each treatment, okay? 1, 2, 3 untreated, 3 Taxol, 3 black mulberry, and 3 saffron. For the 4 of you, each one of you take a treatment. You decide which treatment you're doing, and that's the one that you will be collecting your data on, okay? And you will write the number of blues and number of clears, and then the total number of cells for your particular treatment, okay? So each student will have basically 3 images to score. Once you have done this absolute numbers, you will then take your treatment and convert that into percentages, a percentage blue and percent clear for your treatment for your 3 images. And then lastly, you're going to be calculating the average percent viability and the standard deviation of your treatment, and you will run that. So for these ones, we want you to be looking at, I want you to be using an Excel document to organize that data so that you can to run the statistics later on as well. I'm gonna open one in a second. So this is the one that I was going to show you how to run. Clear contents. Okay, I'll show that to you later. So like I said, all the graphs in there, all the tables in there are already set, so you can just click on that square and copy and paste them into the Excel document. What you should do is to all your put all your absolute numbers in there, and then we are going to be converting them into percentages and average percent and standard deviation, so we can create our graph and run our statistics. I'm going to go over all these statistics and everything else in a little bit when we are at a stopping point in a little while after you collect the data itself. So we'll talk about the statistics and how to present them in a little bit. The last thing you're going to be doing is morphology analysis. For morphology analysis, I'm going to come by your breakout rooms and I'm going to assign you with a sample cell line and a treatment label to examine for your particular group. Everyone is looking at the same image, but everyone is making their own observations. So you'll write your names, and then you'll write what number of cells, what number of nuclei you found in your image, the same image. So remember, it's the same image being looked at by 4 different people. And I want you guys not to be doing this collaboratively, but individually, because I want you to see how those numbers can be a little bit variable, even using programs like ImageJ when different people are looking at them. The next thing you will look at is dead and dying cells or dividing cells and write those absolute numbers. And then you are going to be converting those numbers into percentages. For the top one, you are going to be looking at nuclei per cell at the end of the day. So you will take your number of nuclei. So if I got 90 for my number of nuclei and 30 for my cell, the nuclei per cell would be 3. So you would do those things. And then last thing that you will do is do the biggest cell in the field and the smallest cell in the field. Mark it out and measure it and write that information in micrometers. So those information can be found over here. So for measurement, you need a calibration image. The calibration image is right there that you will use to calibrate your images software so you can measure your cell, just like we did last week. And your assigned image for morphology will be one of the ones in this particular drive. I'll come by and I will give you your image to examine. And so when I give you the label of your image, that's the one that you're going to be looking at. Everyone good? Yes?
I think so.
Very quiet, guys. Are you scared? Are you happy? Are you sad?
Very scared.
You're scared. Okay. There's no need to be scared. So what I want you to focus on, how about that? So I'm going to put you guys in your breakout rooms and you'll have till 10:30. What I want you to focus on is getting your relative growth and your these numbers, the tripe and blue numbers done by that time. At 10:30, we will reconvene and I will actually go through an example morphology image just to remind you how to do all those things. And then you can make those observations. Is that how you would like to do it? Or would you like me to go through the image now? I can go through the image right now as well. I'm just like worried that you guys will, I don't know if I'm talking too much.
I'm okay for waiting to do it after we do this.
Yeah, so let's do that. We'll break it up into little pieces so everyone feels a little bit better. Sounds good?
Good.
Yes.
Okay.
Sounds good.
So at this time, I'm going to go ahead and I am going to put you in your breakout rooms. Keep an eye on the chat. I will write your assigned image in the chat as well. And then I'll come by and I'll check up on you guys and make sure you know which image you're doing as well. But right now you're working on relative growth and the tripe and blue first. Okay. So breakout rooms. I'm gonna go ahead and open the room so you can all go hang out there and do some work.